Thank you very much for that warm and generous introduction. I'd like to thank the organisers as well for giving me the opportunity to speak to this impressive group. So I'm going to talk about how we understand the oral microbiome in both health and disease. So I'm going to cover and emphasise that this oral microbiome is essential for our health and well-being, and we call this relationship symbiosis. And this is in contrast to what we've spent more time investigating, which is the role of the oral organisms in disease. And the term we now use is that this is dysbiosis. But what we need to consider is the transition from health to disease. What causes this relationship to break down and for disease to occur? And if we can understand this, does that have implications for treatment? In other words, can we promote symbiosis and prevent dysbiosis? So just a quick summary of some details about the oral microbiome in health. So the oral microbiome is the collection of all the genomes that are present in the microorganisms in the mouth on all oral surfaces. And any one person can have 100 to 300 microbial species in their mouth. And although we focus a lot on dental plaque and therefore on the teeth, it should be remembered that 80% of the microbiome is found on oral mucosal surfaces. However, this microbiome does live in harmony with us. And indeed, it's essential for our general well-being. And this beneficial relationship we call symbiosis, in that both parties gain some benefit from it. So the microorganisms have a nice warm and nutritious environment in which to grow, and a favourable environment in terms of temperature, gaseous atmosphere, and so on. And in return, they help exclude the sort of microbial pathogens we come into contact with on a regular basis, termed pathogen exclusion. They also modulate the host defences so that, in general, we don't overreact to the presence of these beneficial microbes on our surfaces. And they're also important in the regulation of cardiovascular and other physiological activities. So therefore, in health, we have this symbiotic relationship. So in general, with people who have a sensible diet, an appropriate lifestyle, coupled with effective oral hygiene and effective host defenses, then everything is in balance. The microbe interactions are in equilibrium, and indeed the relationship between these microbes and the host is also balanced. And this is what we see in the healthy mouth. But it should be emphasized that this is a balance. It is an equilibrium, and it's dynamic. And like all relationships, it can break down if not carefully managed and looked after. So in individuals that don't have an appropriate diet, their oral hygiene may be poor, and it's possible that their saliva flow is impaired, for example, due to aging or to the taking of other drugs. The host defenses can be compromised, and indeed people may have lifestyle risk factors like smoking or their diet. Then this alters the relationship between the microbes, and therefore the relationship between these microbes and the host. And a consequence of this is that we often see caries or periodontal diseases, and even possibly more serious systemic diseases as a result of the breakdown of this relationship. And as I said, this is termed now dysbiosis. So just thinking about this in a little bit more detail, we know that in health we have biofilm microbial communities that are reasonably stable over time. We know some of the microorganisms that are present and generally have positive associations with us. And this is maintained in a dynamic environment, as Professor Zaurer just explained. So it may be I shift homeostatic mechanisms to allostasis mechanisms. However, this relationship can break down and we can have dysbiosis. So we know that in patients or sites with caries, we see increases in bacteria that make lots of acid and, most importantly, tolerate the low pH generated from this metabolism. And there's a group of organisms that we commonly see at these sites, like the mutant streptococci, lactobacilli, and other bacteria that make lots of acid. And the properties that are linked to these organisms are fairly general. So it's making acid, tolerating acid, and the production of intracellular and extracellular polysaccharides from sucrose. In contrast, in periodontal diseases, we see a change in the microbiome, but to a different collection of microorganisms. So we increases in gram-negative anaerobic bacteria. So we see increases in spirochetes, in organisms such as Porphyromonas gingivalis, and other organisms that are present are difficult to culture on their own in the laboratory. But many, many others are being recognized now. Again, the traits that are associated with these organisms are fairly generic. They have a proteolytic style of metabolism, they stimulate inflammation, and indeed they're able to subvert the host inflammatory response. So we have an exaggerated inflammatory response, and they also produce cytokines that can damage the host cells. And the question is, we now know quite well the types of organisms we found in each disease, but what we are less clear about is what is driving these changes away from a symbiotic relationship with these organisms to a dysbiotic one. And indeed, where do the pathogens come from? Because if we could understand this, we could perhaps start to intervene and do something about it and push the situation back towards a symbiotic relationship. So, where do the pathogens come from? So let's simplify things. Microbiologists like to give difficult names to lots of organisms, making it pretty difficult for non-specialists to understand. So let's call all the beneficial organisms, just show them as green, and the potentially pathogenic organisms as red. We know that in disease, whether it be caries or periodontal disease, we get far higher numbers of the red, potentially pathogenic ones, than the green beneficial ones. But where have these organisms come from? How do they suddenly dominate at a site? There are two main schools of thought. One is that they are present in health, but at extremely low levels that are clinically insignificant, or we acquire them by some process of transmission. However, for these few organisms to suddenly outcompete bacteria that are beneficial to us and with which we've co-evolved for many millennia, something major has to happen at that site for these organisms to suddenly become dominant. So how does symbiosis break down? Because if we can understand this, we can start to provide new opportunities to prevent dysbiosis and promote symbiosis. So if we look to see where the pathogens come from, generally people always look at the microorganisms you find in disease. But if you also look at the control sites, so those are by definition usually disease-free, you can often find the so-called pathogen present in health, but in low numbers. And here we see studies of periodontal disease, where bacteria are shown in the left panel in health and in the right-hand panel in periodontitis. And if we look at the so-called red complex, a group of organisms that were linked to periodontitis, we can see that they are certainly present in higher numbers and more often in periodontitis, but they were still detected at sites with health. So the interpretation of this is that the implicated pathogens in caries and periodontal diseases can be detected in health, although they are present in lower proportions and less frequently. So we still have to answer the question, how does symbiosis break down and dysbiosis occur? So these organisms are suddenly able to escape from the homeostatic or allostatic factors that control them normally in health to suddenly be predominant in disease. The original hypotheses to explain the transition from health to disease were either the specific plaque hypothesis, which was a major advance in our understanding because it said out of all of the organisms you can see in dental plaque, disease is down to the activity of just a few of these. And you could start to focus in on more studies looking at the properties of these organisms. This was contrasted with the non-specific hypothesis that said disease will really be the outcome of the interaction of both the beneficial ones and the disease-associated organisms. there were certain implications or consequences from whether you're an advocate of one hypothesis or the other. So if you were a specific plaque hypothesis person, then by definition, you could follow up by saying disease could be prevented by specific interventions, such as antimicrobial agents and vaccines. Whereas if you were more in the non-specific camp, you might think about more generic approaches, such as plaque control and fluoride. But neither hypothesis really explained why disease occurred. In other words, why did symbiosis break down, leading to the increase in these organisms and disease? So this led me many years ago now to propose an alternative viewpoint that built on the strengths of both previous hypotheses, called the ecological plaque hypothesis, which intended to bring in the concept of the role of the environment in driving the dysbiosis. And there have been several variants of this, the extended caries ecological plaque hypothesis and the polymicrobial and dysbiosis model, particularly in relation to periodontal disease. But the implications of an ecological understanding to dental diseases is that disease could be prevented or controlled not only by directly inhibiting the causative organisms, but also by promoting the natural balance, in other words, the symbiotic relationship, and interfering with the factors that drive dysbiosis. And specificity in disease was not so much in terms of microbial name, but actually in terms of the microbial function. So, for example, bacteria implicated in caries will generally have the properties that they make lots of acid and they tolerate the acid produced. Other features of the ecological plaque hypothesis was that the etiology need not be monospecific. Any species could contribute if you had relevant properties. You could also carry the so-called pathogens, but at clinically insignificant levels in health. And as I said, disease is preventable by not only inhibiting those organisms, but by maintaining the natural forces that keep the symbiotic relationship, and most importantly, by interfering with the factors that are driving dysbiosis. And it emphasized the important role of the environment as a driver of dysbiosis. So how might this work in caries? So if you look at the left-hand panels, you can see that in health, you either have a few of these potentially cariogenic organisms, or you acquire them from someone you're in close proximity to. And we know that if you have caries, you've got lots more of the cariogenic organisms than in health. We know that the organisms are often mutant streptococci or lactobacilli, and now we have other organisms like the bifidobacteria and Scardovia species. But for these organisms to suddenly become predominant, I've argued that there has to be a major ecological change at the site. And the most likely factors are the frequent intake of fermentable sugars in the diet, or a reduced flow of saliva, and the generation of acidic pH in the biofilm for prolonged periods. Because the low pH alters the bacterial competitiveness of the organisms in the biofilm, favoring those we associate with caries. And the key functions are making acid, tolerating acid, and producing various polysaccharides. So if we want to prevent caries to keep on happening, we have to identify what the pressure is in that patient and interfere with those driving factors. So then we can try and push the system back towards a symbiotic relationship and prevent further episodes of dysbiosis. So in gingivitis, again, we know that there is more plaque found at sites with inflammation, and again, we get more of the potentially pathogenic organisms. And again, the scenario may be that these organisms are present in health, but in low numbers, or we acquire them. We know that the organisms are often obligately anaerobic, they're pro-inflammatory, and indeed they've now been described as inflammophilic. They actually like the conditions of inflammation, that favors their growth. And there are too many species to list here. Again, my argument has been that in order for these organisms to predominate, some major change has to have happened at the site of disease. And the most common things are an increased flow of gingival crevicular fluid, which not only brings in components of the host defenses, but also many host proteins that can act as sources of nutrition for some of these fastidious organisms. So we have increased inflammation, perhaps reduced immunity at the site, and again, local changes are an increase in pH and temperature. The consequences of inflammation are, again, it can alter the competitiveness of the bacteria in the subgingival biofilm. And the key functions of these organisms are being proteolytic, anaerobic, capable of subverting the host defenses, and being inflammophilic, actually liking the conditions of inflammation. Again, if we're going to stop episodes of dysbiosis, we need to recognize what the factors are and try and interfere with them so we can push the system back towards symbiosis. Because, again, the consequences of inflammation are driving this change. So how is it possible to start to alter our biofilms? So good mechanical plaque control will be fundamental to any stratagem. But how else, in patients who are less fully compliant, can we maintain the benefits of our oral microbiome and prevent dysbiosis? So some people are looking at, can we manipulate the composition of the oral microbiome? So they're looking at probiotics and prebiotics. Prebiotics being factors that can be utilized by the beneficial organisms. So this gives them a boost. So in terms of probiotics, there's been some streptococci recognized recently that are naturally found in the mouth, particularly in people with a low caries experience. These strains are both inhibitory to organisms such as mutant streptococci, but they're also arginolytic in that they can break down arginine and make alkali from them. And studies in Belgium are again screening a range of compounds, and they found some that will promote the growth of beneficial organisms while having a neutral or even inhibitory effect on some of the pathogens in biofilm consortia. Another approach would be to manipulate the local environment, for example, by boosting saliva or the innate defenses. For gingivitis, it could be to reduce the consequences of inflammation. So it's possible to stimulate saliva flow by sugar-free gum or using products with non-fermentable sweeteners in them. And novel anti-inflammatory agents are being explored as molecules that will cut off the deleterious consequences of inflammation and promote wound healing. And a third approach would be to modulate biofilm growth and metabolism, not just on the teeth, but also on the mucosal surfaces, as the mucosal surfaces can act as reservoirs of bacteria for disease. And it's been my contention that if you're able to merely slow bacterial growth without killing organisms, that could be beneficial in a dynamic situation like the mouth. And certainly reducing acid production, for example, using metal salts such as zinc, or promoting alkali production with arginine or urea would again help to mitigate against the effects of acid production. And again, some compounds can have an effect not by killing organisms, but by inhibiting enzyme activities, including proteases. So oral care products are often described as being broad spectrum or containing broad spectrum antimicrobial agents. And yet they're supposed to be compatible with promoting oral health, where we have many beneficial organisms. And I think this apparent paradox can be explained by the fact that when delivered from dental products, oral care products, these agents have subtle and a selective mode of action. Remember, the target is a biofilm, and biofilms are far less susceptible to antimicrobial agents than the same organisms when in free suspension. And also remember the pharmacokinetics of antimicrobial agents delivered from oral care products. So here we have a slide from early work by Professor Zaura, where she looked at a biofilm in the mouth that had been stained with a live dead stain, where viable bacteria appear green and dead bacteria appear red. Chlorhexidine has been applied to the surface, and you can see the effect of that agent, described as being broad spectrum, is really on the outer layers. So its effect, if used regularly, would be just to knock back the biofilm, not eliminate it. And if we think about the pharmacokinetics, we deliver these agents perhaps twice a day after a rinsing or application for just a few minutes. These products will be present in the mouth at high concentrations that could be lethal to bacteria for just a few minutes, but they are present for far longer at sublethal concentrations. And as I said earlier, my contention is that these sublethal concentrations can be very effective in the mouth, where you're taking over-the-counter products twice a day, 7 days a week, for perhaps the rest of your life. Because at these concentrations, they can inhibit virulence factors such as acid production, protease activity, the production of the polysaccharides that make up the biofilm matrix, and they'll also slow bacterial growth. In the future, what we need to see happen is the improved penetration of these agents into the biofilm so they can get further into the biofilm, and to increase their retention in the mouth so they're present for longer at some form of active concentration. So to start to summarize, what we all have is a healthy oral microbiome which deliver essential health benefits to us. We could not survive without these beneficial organisms. However, if we have poor oral hygiene or lifestyle risk factors, then these could include frequent sugar intake, reduced flow of saliva, risk factors such as sugar intake, smoking. We can end up with thicker biofilms on our surfaces. And this increases the risk of caries and gingivitis because we have increased acid production. The biofilm spends longer conditions at low pH. It is far more likely to be inflammatory. And also these conditions suppress the growth of the beneficial bacteria. So ideally, what we want to happen is for people to have effective oral hygiene practices and use effective interventions, so using effective oral care products, because this over time should result in less biofilm and thinner biofilms. And this will therefore diminish the growth of acid production. We would get less inflammation, reduce the risks of halitosis, take away the benefits that the pathogens need in order to outcompete the beneficial organisms, and therefore should maintain the levels of these beneficial organisms. So we need to apply strategies that prevent dysbiosis but actually promotes a healthy oral microbiome. So this oral microbiome, remember, is natural and confers important health benefits to the host and can remain relatively stable over time, but is influenced by and responds to changes to the local environment. So the symbiotic relationship I've explained can break down, and dysbiosis can occur if this biofilm isn't managed effectively, increasing the risk of disease. So in patients, we need to identify what their particular drivers of dysbiosis are so we can intervene with those. And effective oral care may require a paradigm shift towards controlling or managing the oral microbiota, both on mucosal and dental surfaces, and reducing the risk, the drivers of dysbiosis in order to promote symbiosis and preserve the benefits of a natural oral microbiome. We need to treat the causes of dysbiosis, not just the consequences of disease. So I'd like to thank you for your attention and thank my colleagues in many institutions that have supported me during my work. Thank you very much.